What is flame photometer? Flame photometer is an analytical instrument used in clinical laboratories for determining of sodium, potassium, lithium, calcium ions in body fluids. Parts of flame photometer. Simple flame photometer consists of the following basic components. First is the source of flame. A burning in the flame photometer is the source of flame. It can be maintained at a constant temperature. The temperature of the flame is one of the critical factors in flame photometry. Number 2 is the nebulizer. It is used to send homogeneous solution into the flame at a balanced rate. 3. Optical system. The optical system consists of convex mirror and convex lens. The convex mirror transmits the light emitted from the atoms. Convex mirrors also help to focus the emission to the lens. The lens helps to focus the light on a point or slit. 4 is the photodetector. The intensity of radiation emitted by the flame is measured by photodetector. Here the emitted radiation and converted to an electrical signals with the help of photodetector. These electrical signals are directly proportional to the intensity of light. How to use the flame photometer? Here are the working procedure. Number 1. Both the standard stock solution and sample solution are preferred in fresh distilled water. Number 2. The flame of the photometer is calibrated by adjusting the air and gas. Then the flame is allowed to stabilize for about 5 minutes. Number 3. Now the instrument is switched on and lids of the filter chamber are open to insert appropriate colors filters. Number 4. The readings of the galvanometer are adjusted to 0 by spraying distilled water into the flame. 5. The sensitivity is adjusted by spraying the most concentrated standard working solution into the flame. Now the full-scale deflection of the galvanometer is recorded. 6. Again, distilled water is sprayed into the flame to attain constant reading of the galvanometer. Then the galvanometer is readjusted to 0. 7. Now each of the standard working solution is sprayed into the flame for 3 times and the readings of galvanometer are recorded. After each spray, the apparatus must be thoroughly washed. The process occurring during flame photometer analysis are summarized below. First is desolvation. Desolvation involves drying a sample in a solution. The metal particles in the solvent are dehydrated by the flame and the solvent is evaporated. Evaporation. The metal particles in the sample are also dehydrated. This also leads to the evaporation of the solvent. Next is the excitation. The electrostatic force of attraction between the electrons and nucleus of atoms helps them to absorb a particular amount of energy. The atoms then jump to the higher energy state when excited. Emission. Since the higher energy state is unstable, the atoms jump back to the ground state or low energy state to gain stability. This jumping of atom emits radiations with characteristic wavelength. The radiation is measured by the photodetector. Application of flame photometer. First, flame photometer can be applied both for quantitative and qualitative analysis of elements. The radiations emitted by the flame photometer are characteristic to particular metal. Hence, with the help of photometers, we can detect the presence of any specific elements in the given sample. The presence of some group 2 elements is critical for soil health. We can determine the presence of various alkali and alkaline earth metals in soil sample by conducting flame tests, and the soil can be supplied with specific fertilizer. 3. The concentration of Na class and K class ions are very important in the human body for conducting various metabolic functions. Their concentration can be determined by diluting and aspirating blood serum sample in the flame. 4. Soft drinks, fruit juices, and alcoholic beverages can also be analyzed by using flame photometry. to determine the concentration of various metals and elements. Advantage and disadvantage of flame photometer. Let's go first the advantage. Number 1. The method of analysis is very simple and economical. It is Number 2. It is quick, convenient, selective, and sensitive analysis. Number 3. It is both and qualitative. It is both and qualitative and quantitative in nature. Number 4. Even very low concentrations of metals in the sample can be determined. Number 6. This method can compensate for any unexpected interfering materials present in the sample solution. Number 6. This method can be used to estimate elements which are rarely analyzed. Disadvantages. First, in spite of any advantages, this analysis technique has quite a few disadvantages. Number 2. The accurate concentration of the metal ion in the solution cannot be measured. 3. It cannot directly detect and determine the presence of inert gases. 4. Through this technique measures the total metal content present in the sample, does not provide the information about the molecular structure of the metal present in the sample. Number 5. Only liquid samples may be used. Also, sample preparation becomes lengthy in some cases. And lastly, flame photometry cannot be used for the direct determination of each and every metal atoms. A number of metal atoms cannot be analyzed by this method. The elements such as carbon, hydrogen, and halides cannot be detected due to the non-radiating nature. What is photometry in biology? A photometer is a tool used to measure light. For example, photosynthesis is the term used to describe how plants get food from light. Or a photon is a particle of light. Or a photograph is a picture made from light-sensitive film. Which gas is used in flame photometry? Flame photometry employs a variety of fuels, mainly air, oxygen or nitrous oxides as oxidant. The temperature of the flame depends on fuel-oxidant ratio. What is the difference between flame photometry and atomic absorption spectroscopy? AA and flame photometry are similar, but unlike flame photometry, which measures the intensity of the light emitted from an atomized sample solution, AA measures the light from a particular source absorbed by the flame generated from the sample solution. Thank you.